Today we're going to talk about medical school histology basics, muscle. We will talk about various aspects of muscle, a little bit about the function of muscle, the different types of muscle that facilitate the different functions of the body. We'll have some questions about muscle and just some features of muscle to be able to identify the different types and what characteristics of those types that facilitate their function. Thank you. Medical School Histology Basics, Muscle. Hi, I'm Larry Johnson. I'm a professor at Texas A&M University, and today we're going to talk about the different types of muscle. Now, what's the function of muscle, and what are some features of muscle? Well, the function of muscle is to generate contractile force. That's the overall thing that it does. Locomotion of multicellular animals, the beating of hearts and movement of internal organs depend upon the muscles of different types. And we're going to talk about those types. Distinguishing features of them, muscle has a high concentration of contractile proteins, actin and myosin, and sometimes they're arranged diffusely, like in smooth muscle, and other times they're arranged in sarcomeres. Sarcomeres is the functional unit of stratified muscle. And all three types of muscle, cardiac, skeletal, as well as smooth muscle, all of those come from the mesoderm. So there are three different major types of muscle. You've got skeletal muscle, which is a large cell, and the nuclei are on the periphery, not in the center. It's voluntary. That's the one that we can control. It's large cell, multinucleated, and it has striations, as you can see here. Also striated is cardiac muscle. Cardiac muscle is much smaller. They're branched. The nucleus is in the center. But they do still have striations, and the functional unit is a sarcomere. Branch cell, nucleus in the center, usually one cell and one nucleus, but it could have more than one. Smooth muscle cells are involuntary. We can't control them. And they have a single nucleus, and it's non-striated. The actin and myosin is aligned more diffusely instead of in sarcomeres. So if we look at smooth muscle, we see that these are kind of like cigar-shaped cells, and there's a nucleus that is seen. The nucleus is inside the cell itself. So this is a muscle fiber or muscle cell. And if we look at that, we can see individual muscle cells. There's a nucleus of one, there's a nucleus for another one, and you can see the kind of pink cytoplasm. Here we can see cross-sections of them, the nucleus in the center, but you can see the individual muscle cells there, and it is non-striated, as you see there. If we look at the ureter, which has lots of smooth muscle in it, we can see bundles of smooth muscle here and here and here and all around through there. And it's a little more dark than is the connective tissue that you see here underneath the epithelium. It's a transitional epithelium since it's from the ureter. And then this is connective tissue below. There's also connective tissue on the outside. So we have fibroblasts, and then we have bundles of collagen that we see there, which is lighter staining than is the smooth muscle that we see here. Smooth muscle, you can see a smooth muscle bundle and individual cells there. If you look at a higher mag, you can see individual cells. Here's a drawing that we saw, and this is the actual cell. These are nuclei, so these are multiple cells. This is an individual cell you see there, and it's non-striated. If you look with toluene blue, this is the ductus deferens, so this is pseudostratified columnar epithelium and the lumen in through there, but all this is smooth muscle, and we can see individual muscle fibers or cells. So you can see the nucleus in the center of these, but it's non-striated, as you can see. If we look at rat intestine, we can see the mucosa there, which is epithelium that plus lamina propria, the connective tissue, all the way to the muscularis mucosa. There's muscularis mucosa and a muscularis externa. Both of those are smooth muscle. If we look at those a little closer, smooth muscle of the muscularis mucosa and the muscularis externa, and we can even see the myositis plexus that regulates the contraction of the muscularis mucosa and the Auerbach's plexus. These are nerve cells that regulate peristalsis which is a contraction caused by the muscularis externa. And so if we look at that with H&E, this is a muscle on the outside, the muscularis externa, and we're looking at this little box here. And so we can see cross-sections of individual smooth muscle cells, and you can see longitudinal views. You can see longitudinal views of these muscle cells, individual muscle cells with the nucleus here, no striations in these. If you stain it with PAS, which periodic acid shift, it stains for high concentrations of carbohydrates, you can see that the basement membrane of smooth muscle cells is PAS positive, like you see in epithelium. And so we do just see the nuclei here, but you see the PS staining around each one of these. If you look at arteriole, you can see smooth muscle cells. There's a lumen here, these endothelial cells, but we see higher mag of smooth muscle cells. They have these little apical caveoli on the surface that they have there, and you see a high density of fibers, and also you see these fusiform densities, these different densities, and the fibers are actually attaching to these densities to facilitate the contraction of the muscle cell. There's a nucleus, and you can see the high density of actin and myosin in there. And another one, we can see a similar type thing, the densities that are attached to the plasma membrane or inside the cell, and you can see the host of filaments. There are other organelles in through there, but there's a host of actin and myosin filaments in these, and they're not arranged in a certain way in a sarcomere. And so it's called smooth muscle because it's a lack of the periodicity that you see in a striated muscle. Again, we can see some smooth muscle here, and they're being innervated by the nervous system. And here we see the different parts of a nervous system, how they connect, presynaptic, postsynaptic, and a little bit of axon in through there that would have the vesicles inside regulating the contraction of this smooth muscle. Another type of muscle is skeletal muscle, and here we see a muscle. This is an arm, a muscle. We can see a fascicle muscle, which is groups of cells, and then we have individual muscle fiber or muscle cell. So this individual cell, you can see it the same over here with nuclei on the periphery, nuclei located here, a big cell. And if you look inside there, you have myofibrils. Myofibrils are linear structures that are inside the muscle fiber. Muscle fiber here with a plasma membrane, nuclei on the periphery, and then we have inside the cell myofibrils. If you look at the myofibril, you can see some dark areas and some light areas. And you can even see a darker structure there, which is a Z disc here and here. And the sarcomere is from one Z disc to another Z disc. And you can see that this is myosin, and the actin, the smaller filaments, is actin. And so it's the location of these in relation to one another that makes the sarcomere contract, as we'll see here in a minute. So if we look at skeletal muscle, we can see the nuclei in the periphery, and we see some capillaries in between there. And here we see connective tissue, and this is the perimysium. The connective tissue that we see here is the perimysium here, skeletal muscle cells, capillaries in between. This is nerve that we see. Nerve is running through here, connective tissue of the perimysium. Now, individual cells have connective tissue in there. We call it endomysium. In a minute, we'll look at that. But remember, what we see here is a muscle cells are innervated, so they can be more than one cell innervated by the same nerve, and so that's what we see here in the muscle. These are the motor end plates where the nerve is coming down, innervating these muscle cells that we see there. One nerve innervates more than one muscle cell. So if we get back to our light and dark areas, we see the dark band A band, and the A band is anisotrophic. In other words, it does alter polarized light. The A band, that's where it goes A for anisotrophic. Isotropic I band does not alter polarized light. And so it's the lighter band in between. And in between there you have the Z disc. So from one Z disc to another Z disc, is one sarcomere. And you can see that in contraction, what you have is Z discs come closer together. For relaxation or stretching, they're further apart. And so so the sarcomeres are further apart in the case of stretch. In the case of contracted, they're close together. And so if they totally overlap the actin overlaps the myosin, you have it contracted. If there's spaces that you have either actin alone or myosin alone, which is the H band, that is the relaxed state. So we see the dark is the A band and the I band is the light band. So if we look at muscle, here is a piece of muscle. On the outside of the muscle, we see the epimysium, epi outside, way outside connective tissue. But we see divisions inside there. That's the perimysium that goes in through there. And then if you have around individual muscle cells would be the endomysium. So endomysium is around individual muscle cells that are inside there. So the perimysium is coarse connective tissue. The epimysium is coarse, less coarse with the perimysium, but the endomysium is delicate connective tissue around individual cells, and this is where we saw the capillaries in the endomysium. So this is a fascicle of muscle cells here and here, endomysium in between. So these are nuclei of muscle cells, connective tissue of the perimysium that we see, and indeed the nuclei on the periphery, and it's striated with light and dark bands, the A band being the dark band. If we look here in between these muscle cells and the endomysium, we can see capillaries. These are endothelial cells of capillaries. You can see the capillaries branching as they go through there. You have to be a little crazy like this, I guess, but these are individual muscle cells, and you can see in the endomysium, you see capillaries that are feeding these cells. If you look at another view of this, you can see the striations very nicely, nuclei on the periphery as you see there, cross-section and longitudinal section, and you can see the A and I band. And at a higher mag, you can see not only do you have the A band, the dark band, the light band is the I band, but you see the Z disc as well. So you can see the Z disc right in through there. From one Z disc to another Z disc is one sarcomere, and you have repeating units of that along the muscle that contracts. One muscle cell is attached to another muscle cell by the endomysium is what attaches one to another. And so we see electron microscopic view of skeletal muscle cell. Here's a nucleus on the periphery, and you can see the sarcomere from Z line to Z, from Z disc to Z disc or Z line. One sarcomere. The H band is a lighter band where you do not have overlap between actin and myosin. The A band, the dark part here of the A band stays the same. It's the I band that moves. And so here we can see in the esophagus, you have a combination of skeletal muscle and smooth muscle. You can see the smaller smooth muscle cells, the nuclei and periphery of these skeletal muscle cells, nerve, connective tissue in through there. We can see it there. This is skeletal muscle and smooth muscle. If you start at the beginning, at the top of your esophagus, you have mostly skeletal muscle, but as you go toward the stomach, you have more smooth muscle, so that it changes where you're located in the esophagus. This is muscle that's highly contracted, so you can see the Z disc here, and you can hardly see an I band at all. So when it's highly contracted, the I band almost disappears, as you can see there. And certainly the H band in the middle has disappeared. So you can see the hypercontracted of this muscle compared to the other one that we saw. So I mentioned the motor end plates where one nerve innervates more than one. Also, there's a muscle spindle, which are receptors, a stretch receptor, and it has a connective tissue capsule, and then some of the fibers themselves are located. So these are muscle cell fibers that are inside the capsule. So the motor end plates we saw before, innervating the skeletal muscle cells, but this is a muscle spindle. And you can see this is a longitudinal view of it. This is a cross-section of one, but you can see these fibers. If you see a higher mag of these, you can see there's actually striations of these intrafusal fibers that are inside the capsule. The capsule is dense, irregular connective tissue that goes around it. If we look at the tongue, we can see another one. Here is a muscle spindle here, and there's another muscle spindle here, and these are the interfusal fibers right here, and this is the end of one of those fibers. But that's what we see. And this is a stretch receptor in muscle. So here's a skeletal muscle that we see with the light and dark bands that we see. And we saw that down here. You can see the Z disc from here and here. Also, the same thing, you have the striation in skeletal muscle and a cardiac muscle too. But in addition to having those striations, you have a bigger striation, and these are the intercalated disc. This is where one cell is attached to another cell, and you can see there's intercalated disc here and here. Also, you might note that in cardiac muscle cells, the nucleus is in the center of the cell. It's not on the periphery as it is skeletal. So this is the intercalated disc at the electron microscopic level, as you can see. And then it is striated muscle, so we have an A band and an I band, just like before, and the sarcomere is from one Z disc to another Z disc. And so if you look at intercalated disc, you can see them here, here, and here, and that's in addition to the A and I band of the muscle, and you can see the nuclei in the periphery. So we can see one of these nuclei in the periphery. That can see what we see there. These are cells of the endomysium. These are fibroblasts of the endomysium that's there, but we can see these intercalated discs. Now, in this specimen, the cells have broken apart at the intercalated disc, and so we don't see the disc because the disc is broken, but we can see that the cells are branched, as you see here, and they're attached to more than one cell. So in this case, the intercalated disc would be where here you can see a little bit of the darkness of the intercalated disc right there. Also, muscle accumulates lipofuscin. So undigested parts of cells can accumulate in old muscle. So these are muscle cells in through here. This is yellow pigment that we see, golden pigment. Is not, everything is yellow, is not gold. This is lipofuscin. Which is an aging pigment. So if you look at the heart, you can see they have the SA node, the AV node, and then you have these fibers that go down here. This is a bundle of hist that comes down here, and these are Purkinje fibers. Purkinje fibers that causes both ventricles to contract at the same time. Histologically, we can see these Purkinje fibers have very big modified cardiac muscle cells in the connective tissue in between. So this would be in the paramecium that will hit endomysium, be around individual cells, and you can see that these are big cells, much bigger than a typical cardiac muscle cell. They have a lot of glycogen, and it's leached out during the processing, and that's what makes it lighter staining than the muscle cells. So if we look at intercalated disc, we saw it before here and here. And we see the sarcomeres from Z line, Z line. We see the A and I band. As we've talked about before, also we see a transverse tubule. If we look at the next view, you can see in the intercalated disc, there's gap junctions. So the gap junctions don't hold the ends of the cells. The macular adherence and the fascial adherence is what holds the cells together. This is one cell and another cell, but it allows communication from one cell to another. So it allows stimulation to come from one cell to another to stimulate contraction and to regulate contraction, uniform contraction of the cell. So here we can see the gap junction right in through there and a gap junction right through there. And this is the intercalated disc. So in addition to that, another contrast between cardiac muscle and skeletal muscle is that both of these have these transverse tubules. Transverse tubules is the infolding of the plasma membrane. So the plasma membrane infolds deep within the myofibrils. There's a myofibril mitochondria, and the transverse tubule goes around through there. And in the case of, so you got the transverse tubule and the sarcoplasmic reticulum. This is the sarcoplasmic reticulum we see here, which is really the endoplasmic reticulum of the cell. In this case, in the skeletal muscle, the sarcoplasmic reticulum follows the transverse tubule, and anywhere you cut it, you have cut through two parts of sarcoplasmic reticulum, and one of the transverse tubule makes you make three things, or the triad: sarcoplasmic reticulum, transverse tubule, triad. In contrast, the sarcoplasmic reticulum does not lay against the transverse tubule in cardiac muscle, and only intermittently does it interact with it. And you can see, so when you cut it, you just have a dyad, just one transverse tubule and one sarcoplasmic reticulum. Dyad versus triad that we can see. Also note that the transverse tubule is on the Z disc, where in the case of transverse tubule over here is in the AI junction. See, there is the A and this is the I, and it's the AI junction is where you find it. So here we can see skeletal muscle. Transverse tubule is smaller in skeletal muscle than it is in cardiac muscle, and you have a dyad, as we see here, versus a dyad in the case of skeletal muscle, and triad, three components here in skeletal muscle, and you have the AI junction versus here, the transverse tubule is on the Z disc. So a couple three questions that we had: which are true about striated muscle? The A band, dark band, is anisotrophic to polarized light. True. The I band, light band, is isotrophic to polarized light. It does not alter polarized light, and hence its name I and A. True. The endometrium is delicate connective tissue around individual myofibrils. False. It's individual myofibers or individual cells. You have endometrium. The endomysium. The myofibrils are what's inside of a cell, not the cell itself. So the answer is D. Which type of muscle have nuclei in the center and are involuntary? Smooth muscle have nuclei in the center, and they're not under control, so they're involuntary. Yes. Cardiac muscle, one or two nuclei in the center, and they are involuntary. True. Skeletal muscle has multinucleated cells, and they're voluntary. So the answer is D. Which is R related to stimulation of muscle contraction? Motor in place, of course. That's how the nerves tell us the skeletal muscle and the move. Gap junctions, yes. Gap junctions you have in the cardiac muscle cell to allow contractions to occur in synchrony because there's communication between adjacent cells. Purkinje fibers, yes. Purkinje fibers go down through the ventricle to allow both ventricles to contract at the same time. So in summary, muscle function is a function in contractile force. It's got to generate contractile force to move limbs, to move organs, to move things within organs, is why we have the muscle contraction. And you have a high concentration of actin and myosin. Most all cells contract, but certain cells especially contract, and that is the muscle cells. In the case of striated muscle, that makes sarcomeres, or the actin and myosin may be diffusely arranged as in the smooth muscle. So we use smooth muscle cell, individual cells here. We see skeletal muscle cells, cardiac muscle cells, the Purkinje fibers, and this is a transverse tubule on the Z line. This has got to be cardiac muscle. So here's another view of Big Bend National Park. Big mountain over there, and this is where Homestead was. So that's the end of medical school histology basics, muscle. Thank you.